So thank you everyone for coming. Let the last stragglers sit down. So just to remind you which one you signed up to is the data standardization and reuse through public repositories. And I promise you it's not going to be as dry as the title. I don't know what we were thinking. So just as a reminder of what we hope to achieve with this workshop is, I just wanted to mention, so this original workshop was to be organized by Professor Masanori Arita, right here, Claire Donovan, me, head of Metabolomics, the European Bioinformatics Institute, and Shankar Subramaniam, who's the head of Metabolomics Workbench at UCSD, San Diego. SD. Unfortunately, he had a last-minute calendar clash, so Susan Sumner took over. Such a nice person, right there. If you see her during the conference, say thank you, thank you. But actually, the assistant professor in her group, Blake Rushing, is actually going to take all the questions and do the presentation. So even more thanks to him stepping up at the last minute, and for Susan for volunteering him. So already starting well. So, okay, I will then introduce the rest of the panel a little bit later, but I hope that this will be a great interactive session. So this is not going to be a whole whack of talks. The whole point is for you to tell us and all of us to interact. So again, we're going to give a little quick overview from each of our resources about what we're doing to facilitate your submissions right now and to enable your work in the lab. I know you often feel like there's a major disconnect between what you're doing and what you have to upload, but we want to enable you to do that better and understand why you doing it better will really help reuse. Last week I was in Houston at ASMS, and it was amazing how many people came up to us to talk about reuse of metabolomics data in our repositories. Your data will be used if you put it in, and they will give you credit. So it's in your interest, okay, to put it in a good way. Because lots of people write papers about how bad the studies are too. So, you know, think about that. But we'll help you do the best possible. So the other key thing is to gather feedback and requirements from our user communities. We do listen to you. We know there's pain points. We can't make them better unless you tell us what they were. We have people from our teams here, and we will take that feedback back. And then the other key thing is, as there's 3 databases represented here, but there are, I know, other efforts around the world. The Korean Professor Shu is here from South Korea, and she is also establishing a metabolomics repository. And one of the outcomes of this workshop is that we want to set up an ecosystem of metabolomics resources where we all talk to each other in a regional fashion. Okay, so I hope I shouldn't have to tell you that data sharing is really important. You're all scientists. You base your work off other people's work. It's shoulders put on shoulders. Everyone works on existing research. We all love to think we discover something novel, but quite often we're just building and then pushing the field on. It also increases the visibility of your research. It really does, especially since the introduction of DOIs. You can put that, your data set submitted in a repository gives you credit. Just don't forget that. The other key thing, which I've really seen in the 6 years since I took over managing the MetaboLights database, is that the journals are insisting on it. So you won't get your paper out until the work is published in a repository. They have their favorites, that's great. And also, your grant funders. How many of you have seen the NIH requirements for data sharing that were published in January this year? One person. People, you're going to get such a shock when you apply for your next grant from the NIH. Just telling you. Look it up. It is now mandatory to submit. But the key thing is they actually give you money in the grant for that work. So be aware of it. You will get help with this. but you will have to do more than you have done in the past. So FAIR, how many of you here have heard of FAIR? Oh, that's better. Good. Then I won't bore you with doing it again. Just keep it in mind. This is your future. You have no choice. So coming to Metabolomics, my entire career has been in bioinformatics, and I have dealt with just about every omics. And this paper was published a few years ago about what can we learn from the past so that we don't make the mistakes that our colleagues made in previous omics developments. So I want to give you an example. How many of you have heard of the INSDC? Okay. How many of you heard of GenBank? Okay, better. So the INSDC, it's been going for 30 years, almost as long as I've been in bioinformatics. And it operates between DDBJ, the NCBI in the States. DDBJ is the Japanese equivalent. Masanori is the boss, top boss of DDBJ. I represent the EMBL-EBI. I'm not the top boss. I am just the head of the MetaboLights part, but I have been there a long time. NCBI does not currently have a metabolomics resource. The United States version is Metabolomics Workbench in San Diego. But this was set up 30 years ago to exchange raw data feeds of nucleic acids. And the thing about it is, from get-go, they agreed data formats, annotation conventions, and minimal standards for global data exchange. So the key thing is, if you went to GenBank, if you went to ENA in my institute, or you went to DBJ, you would have the same core data from all the sites with bells and whistles for the local communities. But the key thing was the data could be exchanged and it could be queried in the same way. And that was great. And most people never even realized where the data was actually submitted to unless you went to the individual sites. But the key thing over those 30 years was that the field evolved. So back in the day, it would take you a year to get one sequence. Obviously, things changed. So the projects became global and increasingly large. So then it wasn't just individual records, you had entire projects, and they could be global. So 2 new databases were developed, again at each site, bioproject, biosample. Bioproject, whereas you could give all the information about the study design and the project plan and the consortium and all that information, and then all the individual records in the GenBank, for instance, would be tied to that one identifier. So when you were writing a paper, you could just cite the bioproject identifier, and all the individual submissions didn't need to replicate all that information. And then they pushed on again to biosamples. So now you can register one biosample. That sample can be all over the world, and people will refer back to that sample information from the nucleotide record, and you don't have to replicate the data all over again. And this is done at each site. Then things change again, and we got transcriptomics. And again, they developed specific databases for the metadata for that, but the raw sequence data was submitted back in GenBank. Then we had human genomes. And again, JGA, Japan, EGA is the European equivalent, and dbGaP at NCBI. They all have your private human genomes, if you've submitted them. And obviously they don't share, because countries they take that kind of thing seriously. And actually, even in Europe, there isn't just one. It's a federated model, so there's a copy of that database around Europe, and each country has the same technical infrastructure and data standards, so that they can share metadata so that clinicians can get access to that, but you have to go to the individual countries and ask for permission for whatever it is that you're interested in. So why I'm talking all about this genomics transcriptome stuff to you is just to show that this is where we would like to go with metabolomics. That we would like that the repositories all work together to make sure that when you submit, first of all, it's as easy as possible, and second of all, that the metabolomics data that's around the world can be shared. But we're only baby steps. This is our Masanori, Shankar, and I have been talking for a long time. This is our workshop that was kind of sidetracked by COVID, to kick off what you want and what we can do. And the idea is to write a white paper out of this about what we can actually achieve. On board with this? Is this what you signed up for? Hopefully. Okay, and then the next talk is Professor Masanori Arita, who will tell you about some efforts and his database.
Well, I'm Masanori Arita from DDBJ, and I'd like to introduce a little bit about political background for why we need to collaborate. Well, many of you already know FAIR data principle, but how many of you know CARE data principle? Now, FAIR and CARE are simultaneously referred to quite often. CARE stands for C for collective benefit, A for authority to control, R for responsibility, E for ethics. It's about data provenance and data sovereignty. And CARE has been extensively, I would say, extensively proposed by many countries in the global south. And it is to acknowledge traditional knowledge of indigenous or local communities, and also to acknowledge their control rights. For example, think about traditional medicine. For example, in Japan, we use herbal medicines extensively. We have our own local knowledge. India has its own, China has its own, probably each country has its own local traditional medicine. But what happens when international big company or farmer obtains a patent on that knowledge? Well, maybe this is an infringement. So this CARE principle talks about such events not to happen, and that's why FAIR and CARE are simultaneously referred to these days. And you have to actually care about this principle, and because you are not, or maybe you think that you are exempted or irrelevant to this kind of issue just because you are working as pure scientist or basic scientist, but you are not free from this kind of discussion because of these international treaties. One is CBD, Convention on Biological Diversity, and the other one, recently decided, is called BBNJ. Let me talk about CBD first. It's an old treaty determined or decided 1993, and currently 193 parties, mostly countries, have been agreed to this CBD treaty. Its main goals are three. One is conservation of biological diversity. Of course, it's understandable. The second one is sustainable use of the components of biological diversity. Okay, it's rational. And the third, this is important, fair and equitable sharing of the benefits. It's not only referring to monetary benefits, but also sharing of knowledge through capacity building or lecture courses. It's also sharing benefits, but anyway, it tells all participating parties, countries and regions to share benefits obtained or arising out of the genetic resources. Then, what's the definition of genetic resources? So far, we have been discussing about materials only, like animals or plants obtained from each country. But now, since last December, these genetic resources include information too. It was decided in the last COP15, that we need to share benefits arising from digital sequence information. We call it DSI. So it covers databases, it covers actually not only genetic sequences, but also downstream information like protein sequences, and some countries insist on enzymatic functions, and even metabolites are DSI from genetic resources. And it was decided that we have to share benefits arising from metabolites or enzymes. It's an international treaty. And even further, BBNJ was decided last March. BBNJ stands for biodiversity beyond national jurisdiction. And it's a treaty about the high sea, public sea, beyond the national jurisdiction. And if you obtain a genetic material, animals or plants, planktons, all kinds of genetic information, you have to share benefits arising from that information, not only material, but also from information. Again, if you identify metabolites from a material, marine material, for example, and if you publicize that information without any notice or without any care about participating nations, maybe you are in trouble. So it's a very, very important international decision. So, but how can we share benefits, or how we monitor the use of DSI? That's the very big problem. But strangely, this how part is not yet decided. We need to discuss 3 years from now among participating parties. What has been decided is, I quote sentences from BBNJ treaty here. Please read these sentences. Monetary benefits from the utilization of MGRs. MGR stands for Marine Genetic Resources, and DSI, Digital Sequence Information on MGRs of ABNJ. ABNJ stands for, I'm sorry, there are so many acronyms, but ABNJ stands for Area Beyond National Jurisdiction. So it's about the high sea. And so monetary benefits from the high sea, including commercialization, shall be shared fairly and equitably through the financial mechanism established under Article 52. So we have to establish such a financial mechanism to share benefits arising not only from materials but also information. And BBNJ, this treaty also says we need to report at least once every 2 years to the Conference of the Parties on the implementation of area-based management tools and related measures, including marine protected areas. What this says is we need to report every 2 years about the usage or access to the information and material from the high sea. So probably it's the responsibility of databases. That's why we need to cooperate to standardize information and to be able to produce statistical, to be able to produce statistics on access and usage of DSI, digital sequence information. So if you measure materials, whatever they are, plants, animals, soil, water, and if you register information, please clarify the provenance of the data, where it's taken and how it's taken when. This is very, very important, especially for parties or countries participating to CBD and BBNJ. And as database providers, we will need to collaborate and provide statistics every 2 years in the future. It's a really hard task, but we need to find a way to do this. Well, to cope with this kind of political changes, INSDC has stated an announcement last March saying that we will mandate spatio-temporal metadata for submitted genetic resources. So for digital, for sequences, DNAs, RNAs, and also for transcriptomes and others, we will collect spatio-temporal information, geospatial location of your data samples, and also species names. And of course, it will apply to metabolomics because metabolome information is a part of DSI. So for now, I switch to the explanation of MetaboBank, and I will go quickly. MetaboBank is the youngest repository, and it's just started in Japan last year. So far, we are accepting data from basically Japanese researchers, but of course, we accept submissions from other countries, from other areas. Your data are welcome. And this MetaboBank repository is linked with biosample and bio project databases. Basically because we need the provenance of your submission data. So when you submit metabolome data to MetaboBank, please register bioproject and biosample first. It's the same scheme when you submit transcriptome data or genome data. So through bioproject and biosample, we database managers can output statistics of the provenance of data. And after submitting these metadata, you will register metabolome information using the what we call IDF and SDRF data format. If you have an experience of submitting transcriptome data, probably you are familiar with this IDF, SDRF, because these formats have been used for transcriptome information. I will go through these formats very quickly. This is the submission page of MetaboBank. It's already in English. And if you click the submission tab, then you will see the flow of submission step by step in English. So you can follow these steps to register your data. And what are IDF and SDRF? IDF stands for Investigation Description Format. Well, it's about how data platforms, and it's a part of method section. So part of it is overlapping with bioproject or biosample databases, but this IDF is more detailed about your platform description. And SDRF stands for Sample and Data Relationship Format. So it's basically a link between each data sample and raw data format. So maybe your question is, what about data matrix? Or what about metaboset? What about peak identification, list of metabolites? Basically, we are not much focused on that part because we prefer to delegate such processes or integration part to other databases or other analysis tools. We emphasize about data provenance and description of measurement methods or platforms rather than focusing on metabolite names or their structures. Of course, you can submit your data matrix as MAF, metabolite annotation file, But currently, we leave this format very, very relaxed or almost free style, because each researcher has its own way of describing metabolites, different names, different structures, and different features. But of course, standardization is important, and I would like to introduce an important effort in the field of lipidomics, because lipidomics researchers have been coordinating and standardizing submission methods and also description. If you visit the website of LSI, Lipidomics Standards Initiative, they have their own guidelines and also submission checklist before submission. And already some journals mandate registering this checklist of LSI to accept before acceptance of your submission. So if you are interested in lipid names or abbreviations, please go to this LSI website and also lipid maps, associated lipid maps consortium web page. And there you will be able to see how to describe lipid names, for example, for triglycerides or for ceramides and others. And they have there is international standard way of describing structures depending on the resolution of identification, for example, just MS1 or MSMS. Yeah, that's all. And maybe I switch to, okay, next.
So thank you to Masanori for giving you an insight into some of the politics that we have to deal with as, as part of the repository world. But it is also a heads up to all of you to find out if your country has signed up to this. Maybe your civil servants haven't told you yet. Best check. Next, I would like to invite Dr. Blake Rushing to talk to us on behalf of Metabolomics Workbench. He works at a core facility and has the experience of submitting to the repositories, and he has things to share with us, which, thank you very much.
Okay, thank you. Hey everyone. So again, I'm Blake Rushing, and so I'm here presenting on behalf of Shankar Subramaniam's and his group at University of California, San Diego. So I work with in Dr. Susan Sumner's group, which she has a very long-standing working history with Shankar, and so we have a lot of experience working with the Workbench, and especially with our more recent activities through the HERE program, for example. And so that makes me reasonably qualified to stand up here to you today and sort of give you a rundown about the Workbench, how it works, and some of its major properties. Okay, and it'll help if I can advance the slide. Oh, okay. Guess there's just a bit of a lag. There we go. Okay, so the Metabolomics Workbench is also sometimes referred to as the National Metabolomics Data Repository, or the NMDR. And this was initiated as part of the NIH Common Fund Phase 1 Metabolomics program, which initiated back in 2012. So there's been a good amount of time for this to develop over the years. And so currently there's about 2,500 studies that have been deposited on the Workbench, ranging from a wide range of biomedical or other research topics. And so I'll say before I go any further, the Workbench does have guided tutorials on the website to show you, with a lot of granularity about how to upload your data and all the different elements that have to be involved. So I'm going to just highlight some of that in this presentation, but if you, want more detail, then those resources are there and available to you. But the first step in order to deposit any data on the Workbench is that users are required to make an account and register. And so when you do deposit your data, there is an embargo period that gets applied, and the default is a 1-year period. So there, you have 1 year before anyone else can access your data without a special link that only you are given. So this ensures that you can publish your data even if your manuscript isn't out there and accepted yet. So there's no worries there. And that embargo period can be extended further if needed, or you can go ahead and release it if you're ready. But general elements of the upload include the raw analytical data. So this is just straight from the vendor files. You also upload your pre-processed data, as well as any associated clinical or epidemiological metadata with those samples. And of course, everything here is de-identified. So there's no worry about breaching confidentiality with participants in the case of human subjects. And so for the Workbench, there's a good amount of, I would say, flexibility in terms of how you can upload your data and accompanying information. So for our group, we have a lot of core activities with other labs. And so we interact very closely with the study PI to ensure that everything is appropriate in their eyes. And that includes following suit with any sort of data sharing guidelines that PI is subject to for the cohort that they're using in the case of human subject research, as well as confirming that IRB number and confirmation. And in the case of vertebrate animal research, we also ensure that the study PI has IACUC approval and has that number as well. And so again, this is just something that we do on top of the standard Workbench guidelines, just to ensure that those who we're collaborating with are really on board with this uploading process. And so the general constituents or elements of an upload are listed here, which covers the major parts of it. So the first is that there is a study abstract that accompanies every upload. I'll say here and emphasize that the Workbench is, in addition to just housing the data, it's giving enough data to where someone who, from the outside, can take that data and reuse it. And so in order to do that, there's a lot of information that needs to be made available right there with the upload in the repository. So the abstract is very key. So it lets anybody know what the study was about and major study design elements. Contact information for both the uploader as well as the study PI in cases where those are two different people is also included. There's descriptions of the sample prep and an analytical method for capturing the raw data, as well as information about the pre-processing, the specific type and model of the instrument that's used, the name of the operator who collected the data was included. So there's a lot of granularity about how that pre-processed and raw data files arrived to that site on the Workbench. There's also a wealth of metadata that's included with the samples. So, you don't just want to have a pre-processed data file that has sample 1, 2, 3, 4, 5 and not know anything about it. So along with that de-identified sample ID, there's a lot of other accompanying information. So whether or not that sample is a quality control sample, for example, or a specific reference type, what phenotypic group it was involved in, or maybe some other metadata information like, age or gender or, et cetera. So there's enough information to do analyses and sub-stratified analyses if needed. And so the raw data files, of course, are uploaded, usually just straight as the vendor file. Pre-processed data is also uploaded. For us, that's typically a CSV file. And there's other supplemental files that you can include if desired. So for us, we have different normalization strategies that sometimes we'll upload, or just the unnormalized file in case somebody wants to renormalize that data using a different method, they have that option. And so, yeah, again, so file types are typically vendor file types or maybe MZXML files. metadata, IDs, annotations, or in the form of a text file or CSV file, which Workbench can accept. And so also in the case of those IDs and annotations, the MSI level is added to those to give that standard. And there is also the option to add other ontology systems to that file. And so for our case, we have sort of our ontology system that we utilize that sort of goes beyond the standard MSI levels. So our ontology system gives a very detailed evidence basis for our matches. So we have a separate set of labels for in-house library reference standard matches and public database library matches. And so for our in-house library, like we'll denote whether a compound matches to a peak by mass, retention time, and MSMS, or maybe it's just by mass and retention time, or just by mass and MSMS, but not retention time. And then on the public database side, we have varying degrees of whether it matches by mass or MSMS. And so that level of granularity helps when we're trying to compare with other data sets or harmonize with other results, because that gives extra layers of evidence basis behind those matches. And we find that that's really key for these uploads and communicating with other labs. And so we're always advocating for more information rather than less when it comes to that information behind those matches. And then there's other identifiers that we often include with our matches, and this just helps in harmonizing data sets and overcoming that challenge with compound synonyms. So there's the RefMet ID, and this is an ID that the Workbench really pushes front and center. This is a unified nomenclature for metabolite names. And there's even a tool on the Workbench website where you can upload your list of your names, and it will match it with the RefMet ID. So that's a very nice tool and very streamlines a way to get a common set of names for your peak matches. But other identifiers such as HMDB ID, KEG ID, NG key, NG key 14, those are very common IDs that we will use as well. And the more you have, the better when it comes to harmonizing data sets. And so this is, again, just kind of running down the general elements of an online submission. Again, there's more detailed tutorials on that website with screenshots and very step-by-step walkthroughs to give you a much more granular look at how that's uploaded. But it's always helpful to have as many of these elements ready before the submission as possible. So getting that summary ready, including that abstract and other accompanying contact information, and sample prep, data collection methods is really nice to have up front. And for us, since we're collaborating with a lot of labs, we try to get that ahead of time, so that way it makes the upload as seamless as possible. Study design table, again, that's your accompanying metadata or phenotype information of all your samples. And that's a required element of the upload, again, to help with that reuse of the data. There is a requirement that your sample names have to exactly match what you have in your metadata file, so it's not too useful if you have sample 1 in your preprocessed data, but there is no sample 1 in your metadata file. So that is a check that the Workbench will do to ensure that that matches up. And of course, any protocols and your raw data for the upload should also be collected as early as possible to make this a seamless process. And in cases where the upload is very large, say there's a lot of data files, the Workbench has mechanisms to help with those uploads as well. So one thing I will say is that the Workbench does work with you as the uploader very closely if you need to. And they're very good about if you reach out to them, you have any kind of issue, they will find a way to make your upload work. So they're very responsive. And And another thing that I'll highlight here is that you can have targeted data accompanying your untargeted data for uploads. So some cases you may have a targeted panel that you ran those same samples through that you also collected untargeted data. And so with the Workbench, you can upload both of those together and link those two data types for your samples in that way. So you may have a Biocrates kit that you ran, or a cytokine panel that you ran, or even your concentration response curves that you use to calculate your concentrations off of. You can upload those. Metal panels, exposure data, like if you're interested in like a panel of perfluorinated compounds that you ran a targeted panel on. You could link all that with your untargeted data. And that will be very nice when you want to do downstream analyses, or others want to do downstream analyses, linking that targeted information with the untargeted. And so on that note, there's all these different analyses tools that are located in a toolbox on the Workbench. So in addition to housing that data and all that information, you can do certain univariate and multivariate statistical analysis on your data right in the Workbench, which is really nice. And also, if you want to compare multiple studies together, there's ways you can pull in other studies into that toolbox to analyze them the same way. And because the RefMet ID is added to MetaboLights, it's a very streamlined process where you can say, metabolite A was increased in this study and was also increased in that study. Maybe they're looking at similar endpoints, and you can use that to confirm results, or at least just compare across studies. And so there's many different additions that are being worked on, I will say pretty much all the time. So the Workbench is always looking for new analyses to add to the site to add capabilities here. And so one of the ones that almost done. And so just really quick, one of the more recent additions that's been made is the software ADAP, who I have to credit Sushadu with, who's there in the back. So this is a processing or an automated data analysis pipeline that we've been working really closely with Susha's group to develop. And so this is something that's being integrated into the Workbench to help with all the data pre-processing, as well as peak annotations of data. So this is something we're really excited about, and we're looking forward to seeing this down the road as it gets integrated. All right, and with that, I will end here. Thank you.
Thank you. Okay, so thanks very much. I think that was really, really helpful, giving both viewpoints of what he follows the guidelines from Metabolomics Workbench, but also going above and beyond and providing a confidence in the annotations, which we would encourage many more submitters to do. So that was lovely. Thank you. So, okay. So then the next talk, and I'm going to zoom through this because what I'm really is about our resource, but I want to get on to what all of you are thinking about before you leave. So I will go even quicker with our resource, but just to be aware that 4 members of MetaboLights are here at the conference, and we will, at the end of this presentation, I'll tell you the poster and where to find us. So if there's anything you want to know about us, please come find us. Okay, so MetaboLights was originally set up in 2012, like Metabolomics Workbench, at the European Bioinformatics Institute in Cambridge, UK. And from the key thing that we all share is that we are a repository for studies. We also build a compound library, which I will come back to. From the beginning, ontologies were really important, but they became integral to the redevelopment of MetaboLights that we did in 2017, where we made all of this very, very mandatory. And a huge amount of work was done to select the right ontologies. We do not reinvent the wheel. We are bioinformaticians. We do know new tools are developed, or new terms, and we submit them as appropriate. We do have a small internal ontology, which we are trying to get taken up by worldwide efforts to make sure that people around the world use them. This is a huge amount of work. We're happy to share it if anyone wants it. Lots of people, both commercial and other databases, have taken this on board and implemented it locally, and we're happy to share it. So we have also been involved with the MSI since the beginning, but also newer efforts like MQuack. I hope you all went to the MQuack session, which was just before this one. And the key thing here is that we try to keep up to date with whatever's going on, like the lipidomics one. But we really need, and this will come back to the questions at the end of the session, what your priorities are. There's so much to do. We have a lot to do. We need to know what's important to you. What should we target first? So our database is a little bit different to the other two, of course, which is why we're having this workshop, so we can all agree what is the minimum. So key thing that we break it all up into is the descriptors, the protocols, samples, assays, and MetaboLights, and then the files. So it's really, really important for us to capture the protocol information, and we work very closely, like the Metabolomics Workbench team and like the DDBJ team. There are humans on the end of those emails, okay? Please bear that in mind. Four of them are here. So we read every single thing you write to us. We are listening and we are paying attention. And we need this information to make sure it's reproducible. We really need to know that column. We're not doing it to annoy you. I know you have to go back to the lab, away from the computer, and find out what that column is, but you need to do it. But that seconds what Blake just said. Have your study protocol next to you when you come to do your submission. Then we have all the sample information, and it's really broken down. I just wanted to go through this, but the slides will be made available. But the key, and you can talk to any of us, but the key thing is we do ask for quite a lot of information, and we know that, and we appreciate your pain points. But this is really, really important. And we do insist, well, not insist, if you haven't identified any metabolites, that's fine. I was really sad for your experiment, but it's fine on our side. We hope you've identified metabolites and you would like to submit them to us. And we do ask for information around them. We use KEBI. My colleague here from the EBI, Dr. Ansar Malek, is part of the panel, and hopefully he will answer some questions, but we do work with RefMet. So again, there is a mapping, and we would like to make it public at a certain point, but we do map out to HMDB and KEG and all the rest of it. We are an ecosystem of databases. We all have areas of expertise, but the key message of this workshop and any other is that we want to all work together. We do work with lipid maps as well. And again, to second again what Blake just said, as much information as you can give us about how you identify the metabolite, the better that other people can trust your work and reuse it. So our study submission process is in a number of ways. So we have an online guided tutorial. So you submit, and each step that you go through, you get a tick box. You can't get on to the next stage until you complete the first one, second one, third one. So you know exactly how you're doing as you go along, and you get appropriate help messages to let you know what it is that's failing. And hopefully you can solve those yourself. They're intelligible enough. If they're not, let us know. Again, let us know is the key message. We also have, for larger submitters, we have a template system where they can upload the data themselves, and then we process it on our side and make it public. And we also do this for industry. Another thing related to the Metabolomics toolbox, for instance, is we want to make your lives as easy as possible. So it is Rick Dunn will be giving a talk about this on Thursday. It's live now. It's our beta version. Be kind, but please do try it if this is what you're interested in. You can upload your raw data and run it through the Galaxy workflow to process it right down to MetaboLight. We also have an NMR version, which is not yet available, and Tim Ebels and Michael Judge here in the front talk to them about that. They are here. But this is what we really like to do as a repository, is to work with the people in the field who have the expertise and bring that in-house to make it as helpful as possible to the users. And the thing is also, for those of you who are worried about your climate change commitment or your institute's energy bills, all of this is done on the EBI infrastructure. So you're saving the planet, kind of. Okay, so we already know that some of our MetaboLight studies are being reused in various systems, including GMPS, and that actually prompted us to have a grant with them where we're going to reanalyze all the studies that are suitable in MetaboLights. And then very quickly, we are as one of many different resources at the European Bioinformatics Institute. One of the major advantages of that is that we can work with the other resources in our database, in our institute, to do multi-omics studies. And I know many of you, this is the big key thing. So one project I would like to draw your attention to, but I don't have the time to go into it, but please do come and talk to us about it if you're interested, is the Holofood data portal. And this is a European effort where on the microbiome, and there's histology information, targeted metabolites, host genome, the host transcriptome, the meta information, and all of this is split in, there's a portal that points you to where all the information is stored in the appropriate resources. And we think this is a data model going forward, and we'd really like to hear your feedback on it. And then quickly, this is our team. The ones in bold are actually here in person. Naomi and Callum are sitting here in the front. Tom is here, and it's me. And the rest are at home feeling very jealous. But they have other conferences to look forward to. We all get to leave at a certain point. But it's really great for us to come out to conferences and meet all the people who email us. That's really nice always. It always makes us feel bad when you have a bad day and you send us a mean email. We go, oh, they're having a bad day. Not that they're a mean person, because we've met them and they're nice. So our poster is 1028, which is Wednesday and Thursday. So please come and visit us. There's also a poster on Kebi. And like I said, Rick Dunn will be giving a talk about that Galaxy workflow and the science behind it, which will be on Thursday at 11:05. So now we get, oh, this is my key thing as well. So this is just for MetaboLights. Our submissions, it might come as a surprise to you. First of all, exponential growth in the last few years. This is why it's really, really important we get our standards sorted, as minimal standards sorted as soon as possible, because the data is genuinely exploding. The other key thing that I put there is where the data is coming from. So just to our resource, our biggest submitter is actually China, followed by the USA, followed by the UK, and then around Europe. We really want to move, like it's lovely you're all submitting to us and all, but we really would like to move to a federated model or a collaboration across. So eventually, those of you who are nearer to DDBJ can submit there and submit to a UCSD. But first of all, we have to convince our funders around the world and the journals around the world that we are actually a coherent ecosystem, because right now they play favorites, and we have no influence on that. So we would like to convince them we're all playing fine and you can submit anywhere and it's great. You'll get the same quality and the same commitment. And that's what I would like to achieve. I am a data sharer. This is my thing. I don't need everything on my database. It's fine. Okay. So one way we would like to do that is reinvigorate the old metabolome exchange. so that our databases can continue to submit there, but have the data in standards that you can all query it in a more sensible way than you can right now. So, the exciting part. So we'd like to ask you a few questions. So if you, something went wrong there. Okay, so first of all, can you open your app, please? Cool. So if you select the agenda, if you haven't chosen this already and you have it in your My Agenda, that's even better. But if you haven't, please select Working Group 12. And then if you go right down to the bottom of the screen where your eye does not naturally go, click in the area next to Poll. Yeah, and Cal, it started, so you should see something now. Do you see something? Do you see the question? Anybody? Yep, cool, good. Because I can't see anything. I'm looking at you, so give me some feedback here. That's great. So if we go back to the first question. So what you should see is how do you generate the data you submit? And I apologize for my grammar. I was quite jet lagged. Okay, so and it can be, we set it up that it should be, you can click as many as possible. Please let me know if that's not the case. Is it your own? Is it collaborative laboratories? Is it core facilities, commercial companies, or kits? Are you clicking? While you're clicking, I forgot to introduce the panel. So you've met Masanori Arita. You've met Blake Rushing. Then we have David Gall, who's head of the core facility at Georgia Tech. We have Dr. Thomas Payne, who's the head curator at MetaboLights. We have Dr. Adnan Malik, who's responsible for the KEPI resource. So they all come, and Dr. David Gall is particularly interested in environmental marine stuff, as well as many other things. So we tried to get a diversity of people here on the panel to respond to your questions. So do we think we have enough people answering this? How many? I think we're doing good. 88 people, thank you. And what was the highest result? Okay, you're an own house type of crew. That's good to know. Is that your, just to point out, there are 2 microphones there, and if you have any comments, please use them. And when you do, please introduce yourself. We'd like to know who's talking to us, just as a thing. Okay, so how many people, is that the default position to do in-house stuff, or is it because you don't have the money to go elsewhere, or what? Any feedback? You all want to do your own thing? I see lots of nodding. Individual scientists. Okay. How many of you are part of collaborations where you do the Metabolomics and someone else does something else? Okay, that's good. So you're the kind of people who need to be talking to us about those multi-omics studies, because you all need to talk to each other. Because what we see in collaboratory laboratories is a lot of them who come to submit to us don't have a lot of the metadata, which is with the genomics people, for instance. And that makes it difficult to complete the study. So that's a learning curve that we've seen with people. Okay, on to the next question, please. So we know that there are barriers to submission. We have seen an explosion of submissions in the recent past. But we do know that there's still a lot of data out there. So these were some of the barriers we thought of as what's stopping you submit. So the first of these is the knowledge of what's needed. When you come to submit, have you not thought about what it is that the databases require? You've got a snapshots of each of our resources, which are slightly different in what they ask you. Then the second one is ability to access or generate all data required. So like I said, with some of the collaborations we've seen, the phenotype data, not all of it has been shared with the Metabolomics lab. They've just been sent the samples and said, do this, and they don't have the metadata associated to it. Usability of repositories. If you think we're being hard on you, just click that button. We need to know, we need to make it better. And then knowing that, we can also ask funders to give us more money to make it better. I have no problem doing that. Then there's the lack of reporting standards. As Blake highlighted, MSI standards, internal ontologies that they use, which is better to give more information. Is that something that's holding you back? That you don't know what to put in, again, and then data privacy. So again, Blake highlighted the point that all our databases right now require anonymized data. There is no controlled access version of MetaboLights or Metabolomics Workbench right now, as far as I'm aware. So please click your favorite, or all of the above. Is it done? Okay, and what is the result? No surprise, but thank you. A little more feedback than that would be nice. Please pick up that email to any of your repository of choice that has given you that opinion. Would anyone like I said, this is not a hostile environment. We all want to know. Would anyone like to stand up and let us know what in particular is the problem? Just an example. I promise we'll be polite. Please do stand up.
Hi, I'm Bob Powers, University of Nebraska. So common problems that I or my students have had, and I don't want to pick out a particular database, is more in the technical issues. So in the process of trying to upload data, things would time out, things would crash. Also, there's a lack of response. We rarely ever see email response after you have problems. And also, even if it appears to be a successful upload, a lot of times you don't get any acknowledgments, and then when we go back looking through the database to find our data, it's not there. So, so these type of issues has kind of resulted in difficulty getting people to upload data because it's been 2 weeks and nothing happens. So at least that's my personal experience we've had.
Now, that's very valid. So at least in our database, we are trying now, the files are getting bigger and bigger. So we are now expanding the technology that you can upload and the bandwidth, and also to send automatic messages to say that something has stalled, and to check checksums on either side to make sure what you thought that you sent actually arrived on our side. But that's a valid point that all databases should take into. I hope it wasn't us who didn't answer, but if it was, I apologize. I think we are on the side of over-answering rather than under-answering, but everyone can have a bad day. So thanks for that feedback. And all of this matters because, for instance, that I can take back to the technical infrastructure in my institute, if that was our database, and say, I need more resources. Look, the users are complaining. This is the key thing you all need to understand. While you think complaining doesn't make any difference, it does actually make a difference. It gives us the information that we can to interact with our peer groups at the institute to make our service better. Susan?
So we've had a really great experience with Metabolomics Workbench and with uploading all types of data. It could be untargeted, targeted, and it can be data types that they hadn't really planned for, but then we'll work with them and they'll figure out how to accommodate us. So everything's been a really great experience. I did want to say that the data that we haven't uploaded isn't because I don't want to upload it, but it's because we may be working with a cohort that has their own policies and their own data sharing plan that they worked out with their funding agency, like NIH, at the time that they had the cohort funded, and that cohort doesn't want the data uploaded. So that's one thing. So I'm hoping with the new NIH data sharing rules, I should say. I think they're not guidelines anymore, but maybe
No,
rules.
they're mandatory.
With those rules now, that this may get some of the larger cohorts to start sharing the data. I also wanted to mention that there are independent programs, like I'm in the NIHS Human Health Exposure Analysis Resource Program, and they have their own Workbench, where this data goes. And then in the Nutrition for Precision Health study, that's a new NIH Common Fund study, all of our untargeted data, which is going to be something like maybe 54,000 untargeted data sets and another 20,000 or so targeted data sets, they're going to go on to the All of Us Workbench. And so this is all driven by these large-scale consortia. And so I kind of wanted to mention that. So if there was some way that these resources could start working more closely with the large-scale consortia. But the question I do have is, you had mentioned that we all need to get for the new NIH rules, which is great. I wanted to put that back at you and say, well, what are the rules in Europe, and what are y'all going to do, and what are the other countries going to do, and why is this always focused on what the U.S. is going to do?
Okay, so the European Union is even tougher than the NIH, and they've been tougher for a while. I think the reason why we highlighted it is because the United States is just next door. And I just came from ASMS, where it was a big conversation. There was an entire workshop, and there was 3 officers from the NIH there to try and explain to people how important it is. So I thought it was relevant for this. I'm not quite sure. I know in Europe, it depends on the country. I know Europe is kind of small to those of you who are from America, but there are individual countries, and they all have their own rules, and that's another navigation that the European Bioinformatics Institute has to follow. But there is one major funding authority, which is the European Union, and they have very strong requests. But I would just like, Jules, Mr. Man. I just ask, just to complete the story, I would like to ask Masanori what the rules are in Japan about sharing.
Well, Japan is far behind. We are starting to how enforce publication with embargo, not immediate. So, and it started, I think, last year.
Okay, great. So we're at different points. Jules?
I guess one challenge around the usability of repositories is that you do require somebody that's reasonably well trained, and also that we get this data sort of right at the end often as a project. So if you've had a postdoc working on a particular grant, they might be moving off that particular project, and then how do you go about sort of doing that? So I wonder whether we could learn from open access publishing, where, at least in the UK, we had some block grants that came to the institution that allowed us then to make papers open access, whether we should be lobbying funders to have something similar. So there are grants there, so people, if it's 2-3 months after the end of that grant, you've still got some money where you can employ somebody to make sure that data gets out there.
Yeah, no, I think that's really, really good point. And that's in fact what the NIH has put into their data sharing mandatory guidelines, is that when you apply for the grant, you can apply for specific money at the end of the grant to formalize all the data and upload it. And we do hear that quite often in the emails to MetaboLights. It's like, my postdoc is gone. I don't know what they did or did not do. Here's the data. And it was like, well, that's called supervision. But obviously not. Okay. But so that comes back to training, and that comes to another question that I'd like to move on to. So to barriers to reuse. So what I think it's the same for all of the 3 repositories. Well, you're new. For the MetaboLights and MetaboLomics Workbench, the driving force at the beginning was as a repository, and also to a certain extent as gold standards. So here were examples that people could look at. And at least in MetaboLights at the beginning, there was also a key part, which was that you would have those gold standard studies there for people to develop computational tools on and algorithms. But over time, and especially in the last couple of years, what we have seen is that more and more people are reusing the data. So if you search for any of our identifiers and see them being quoted in papers, they're not being quoted as we deposited the data in these resources. They're being quoted as in we took the data from these repositories and we did something new with it. And that's absolutely great. But any of you here done something like that? Downloaded? Oh, cool. That's great. Thank you so much. That's good. So when you, and I know some the answers to some of this, which is why I wrote those questions, is barriers to reuse. So we have identified data completeness, the data formats. This is fairly, behind that one, I would like to highlight that obviously we have the raw data and we have certain kinds of derived files, and we know that some are easy to use and some are not, but that's the kind of feedback we need to drive forward the community. Data complexity, as Masanori mentioned, the different data, people naming things. We use Kebi to standardize all our names. Metabolomics Workbench use RefMet. But we do want to put cross-references to other things, but it's not wonderful at the moment. The other thing, search functionality on the repositories. Can you find what you're looking for when you come? And training and use cases. So please go to the next question on the app if you have access. So can I ask the 2 core facility people here, Blake and David, do you ever look at other people's data in the repositories?
So I can go first. Yeah, so we do. And we do that for a couple of different reasons. One is to just see what other people are reporting and sort of their standards that they're using. Sometimes we can get better ideas about how we can report our data. And also just get sort of a feel of kind of where we are in relation to other people. And the other side of it is if we want to look at the science, right? Mm-hmm. So maybe we do have similar studies that we're doing. We're doing a lot of very large scale population-based studies. So it's really great when there are other studies that have been uploaded. Maybe they're experimental model systems, or maybe they're just other cohorts. It kind of allows us to, if we want to investigate, any kind of validation, we can see what's out there. So it's really helpful for us. And I will say a third one is also because we have activities in building new software. Again, I'll call out Susha for that. So, another way that we can reuse data is to put it through those software pipelines, because we don't want to develop software just for our data. We want to develop software that can be used for everyone's data. So that's another really important way that we can use that. And both raw and pre-processed data is very useful for that.
Great. And David?
So for most of the time, when we go through and look at a particular study, Yeah, if there was something that we could look back on that other people have done, that would be very useful. I can't say that we've experienced that quite yet, but I've had something that a little bit different, not necessarily that we got a data set from a repository, but I've had collaborators give me a data set that somebody else collected. And if you haven't ever had the experience to try this, you find that you come up with all the problems of why we're talking about all this right now, is because when I got the data set, you think, oh, well, everybody kind of collects it the same way, so I'll try my pipeline and we'll see how far I go. I didn't get very far. I didn't expect to see that the blank file had a different size than everything else, meaning that the runtime was different. That is something that I don't know how to fix out without just eliminating the blank file, which then, of course, this particular one was looking for a particular needle in a haystack. So now I don't know what is real and what's not real. It becomes a very manual problem, and it was something that I had to unfortunately talk with a collaborator and say, look, I'm not going to be able to do too much with this data. We're going to have to recollect because the tools that I have is really going to be a lot faster and a lot more time efficient and cost-wise to do it that way, as opposed to me trying to do this manually over thousands of features and trying to do it that way. So I can definitely talk to the nicety of having all of the metadata that's with each of these things, because if you do want to try to reuse somebody else's data, a lot of this is very important because it's not just the basics. It goes to really understanding what was done, because very quickly you're going to start to have questions that I see something, is it real? And if you don't have the metadata, you really can't address that one way or the other.
That's really great. So has people in the audience also have similar experiences?
So in terms of Metabolomics Workbench, I do know that the access is on the order of millions per year, but the downloads are on the order of thousands per year for people downloading data sets and looking at them. Now, one of the things that I think is really going to help facilitate the downloading and using this data is the ADAPT software that's now on Metabolomics Workbench. And some of you may have attended Dr. Sushadoo's session yesterday, where she was giving a hands-on demonstration of how to use ADAPT. One of the reasons why I think the software tool is there's many reasons why Susha can explain this better than me, but one of the reasons is it can handle very large-scale studies, and much of our software nowadays is very limited to being able to align, thousands of spectra, because the software are cumbersome, I'll say. But ADAPT is very seamless to use and can handle these very large-scale data sets. So I think we'll see more use, even though Metabolomics Workbench is at thousands of downloads already. I think it will even increase more now.
Now, that's great, because you lead me into the next question. But before we move on to the next question, can we have the results of the previous one, Cal? Data complexity. So it's the diversity and complexity of the data that's hindering you. So I'd like to come back to that question, but because we've had such a nice flow into this question from Susan, I'd like to do this one, which is tools and workflows. So we have heard a little about that one, and what we highlighted from Rick Dunstock, which will be on Thursday, is about reuse. So there's two things with that new workflow, is that it will reuse the existing studies in Metabolomics Workbench, and you can upload to that tool Metabolomics Workbench studies and MetaboLights studies, but you can also use your raw data. But we did learn during that process that you need to have specific information in the studies in order to do that. But coming back to the tools, please answer what is holding you back. So I need open source tools. I need workflows that integrate specific tools. And please reach out to us and let us know what tools that you would love to be part of a workflow, because we're writing grants and we can include those in things going forward. I need training and documentation. I'm not a software engineer, but I know they're not great at the whole documentation thing. And I'm looking at you, Cal. Yeah, you wrote it this week. Michael, don't you laugh at him. They have written it now, so you're all good. But it is often the thing at the end, after you've done all the cool thing of working on it. And the fourth option, which I think is not possible, but maybe there are people out there who have all that you need in the area of tools and workflows. If you have, please let us know, and we'd love to know what you have. Okay, please complete the poll. So if we come back to the complexity of the data, I'd like to ask my colleague, Dr. Adnan Malik, about what feedback he would like to give to the metabolomics community from the point of a chemist, and how he thinks we identify metabolites.
So we work quite closely with MetaboLights. So we take a lot of the species data from them. For example, if a metabolite has been detected or isolated from some living organism, so we take that data. We also take structures from metabolites. And the whole process of KEBI is to make sure the structures actually match the names provided, because sometimes you have names which do not match the structures, because chemical structure complexity is quite complex. Structures can be really complex. You can have R enantiomers, S enantiomers, you can have double bonds in different configurations. So we make sure the names actually match the structures. And then in KEBI, we also provide links to other databases. So for example, we provide links to around 65 different resources. So we're increasing the interoperability between the different, not only the biology resources, but also the chemical resources. And our data is then also submitted to PubChem. So via metabolites, so the data that's in metabolites comes to KEBI, and then from KEBI, it goes into PubChem as well. So your data becomes open to other resources as well.
Yeah, that's what they do. So basically, they double check all your identifications based on the hardcore chemistry that many metabolomics, well, not many, some do not have. But it's a really nice check for us when we submit what you have submitted to us to KEBI in order to confirm what it is. Okay, is the poll? Okay, so basically you need more tools. Would anyone like to stand up and give us an idea of an area where you have identified a gap? Come on, people. We know everybody said so. You don't want to say? Yeah.
Is this for reusability of the data or for any specific type of?
Yeah. So, I mean, when you get your data, what are the bottlenecks for you processing it?
But not specifically for reusability.
No. Because it's also for the submission. So when you have the results of your experiments from your mass spec, what are the things that are hindering you from putting it together so that you can complete a submission? Processing the data, the formats. Okay, obviously you don't want to highlight, but you have said in the poll you have issues, so why don't you find one of us at the conference and let us know. We're here for the full time. Or send us an email, any of the repositories, send the email. We will be collating this information afterwards, and we won't be naming names. Don't worry, I don't know what they, just let us know. Because the thing is, then we can make it better for you. As we got funding, both from the BBSRC and the NSF to develop these tools, Metabolomics Workbench has got funding and has collaborators who develop these tools. So if you let us know what you need, then we'll do our best to provide it. But if you don't let us know what is blocking you, we can't do anything about it. Okay, on to the last question. So as I mentioned at the beginning, there are so many areas where metabolomics has gone into in the last couple of years, and there aren't standards for them yet. So unfortunately, we're double booked with the epidemiology workshop, for instance, and this is a community who already knows that they need new standards and working with repositories. So again, all are the above. Which are the areas where you think the repository should focus their standardization efforts with the community? So agriculture, clinical data, epidemiology, exposome, toxicology, or all of the above. That won't help us much, but as in prioritizing. But it's still good to know because it will enable us to reach out to the right people. When you're ready. The rest were? Tied. Okay. Well, thanks. So we will have a lot to do, which is good. Can anyone please, if one of these is your area, can you let us stand up and let us know? If you don't want to stand up, we can bring you a microphone. Let us know what in your area needs particular work. Just an example. Delighted to meet you.
Sorry, there's a long walk up. Hi, I'm Anastasia Pramalena from the Pacific Northwest National Laboratory. I work with standardizing soil data, so I guess agriculture fits in that. And something that we've been struggling with is sample prep, like standardization, which, especially for soil data, I think at ASMS someone said, like, if you're trying to do soil metabolomics, don't. I don't have really a solution, but I have some suggestions that are just kind of being encompassed in our own internal database, but obviously want to extend that to the greater community. I'm going to commit a faux pas of like offering a problem without a solution. That's kind of where we're at.
No, stay there. Paul, David, do you work with soil or is it purely marine?
I mean, I work in a lot of different areas, so.
Yeah, so in the particular area of soil metabolomics?
I mean, we don't have very, I mean, we have a few projects that have come in that regard, but we're not fully indulged into any one particular area. But I mean, I think the concept of sample prep and how everybody does everything plays an important, as everybody in this room knows, it plays an important role in what you see. So it's important that we not only collect that, but then understand how we could add to, you know, if a further future experiment of what adjustments can we make that we may have a slightly different view. How do those two views come together to formulate better information? In this world of complex, untargeted data, I think it's really hard to a priori know exactly what you're going to get until after we run, and then we're trying to combine things together. I don't envy the soil community in this process because there's a lot of factors that were talked about yesterday that I think a lot of people are not quite aware of, and how best to go about and really pull out the things that are going to be measurable. I mean, that's where it all comes down to. I think it's still, in my opinion, I think there's still a lot of interesting work to be done to know what is going to be the best method. I mean, at least yesterday, I think I heard that HF, treatment with HF to release the metabolites off the minerals, that doesn't seem all too appealing to anything that I might want to do, nor acid hydrolysis or base hydrolysis. So I'm hoping that there might be an additional alternative. But again, I think that's still stuff to come. But I think we still need to find a way to share what's been done so that we know what people have done and what we can try next.
If I can comment on that, I think
Yep.
a big issue we've been running into is like standardized ontologies. And just because soil requires so many different words to be used and communicated in a very specific way, that doesn't really apply to a lot of other fields. But if we can leverage existing ontologies, or we've been trying to do, but obviously there's some techniques that just aren't encompassed in that.
Yeah, no, that would be a really nice project for, at least on metabolite side. So it would be great if we could follow up and we could see the terms. Because of the institute we're at, there's a lot of ontologies across really, really diverse systems. So we could either direct you to the right one or introduce you to the people who need to expand their ontologies to include your terms, which was what I would hope would be the case. So please follow up with us afterwards. But that's, yeah, it's amazing how Metabolomics is expanding into new areas and providing a new challenge all the time. Jules?
I just picked on epidemiology. You know, I could have picked sort of different areas, but one of the things that sort of struck us in terms of trying to report our epidemiology data is there's a lot of downstream processing that happens, you know, in terms of how you set up your GWAS analysis, how you do that subsequent sort of analysis then on top of that, things like Mendelian randomization and things. We're not really capturing that sort of in the database. You know, so I don't know whether it's actually to capture it within the Metabolomics database or better linking through to the epidemiology databases, because of course we're also depositing the data, or the data has also been reposited around our cohort as well. So it's about how you get those better links and report in the metadata where those links are as well.
Yeah, so I'd like to ask Thomas Payne to refer to that, because I think what I put up that quick slide about holofood, that I think that would be a good example.
Yeah, that's a good example. Actually, metabolomics data transformation is poorly described anyway. When you talk about even producing your data tables, a lot of people won't provide that information. So then when you're talking about your downstream analysis, once you have your data tables and what you're doing with that to integrate, yeah, that's even less, right? There is a place for it in MetaboLights, for sure. You can put it in data transformation protocols. You can have subheadings, like we ask submitters to provide as much information as possible from your pre-processing parameters through to your statistical methods. So we could see a place for that. And in the multi-omics domain, yeah, it might be in the future that there is somewhere better for that in the future, but for now, we definitely see it in the protocols and the data transformation.
Okay, so that was our final question. So thank you all for participating. Are there, oh no, please, we have plenty of time.
Hi, I'm Kumaran Bhaskaran. So I'm working for Biological Magnetic Resonance Database at UConn Health.
Oh,
so I would like to comment on the data policies. I mean, so most of us talk about the FAIR principle. So we all try to address the first part, the findable data and accessible data, but when it comes to the interoperable and reusable, so we don't pay much attention to it. We leave it to the user's hand, so you download the data and do whatever you want. But we have an experience for the last 2 decades doing the macromolecular NMR spectroscopy. So we tried to do this in the beginning, but it didn't successful. So we were so strict on collecting the metadata. We get so many complaints from the users, so you were asking too many questions.
Yeah.
For deposition, because otherwise the data is not at all useful. So how do we address this? Because compared to the BMRB, what we talk about here is heterogeneous data. So you cannot annotate an heterogeneous data by a single group or single person, because if you go further into the field, it might be integrative methods, not just mass spec. So you can combine mass spec and the NMR, and some other techniques may come in the future. And all these things, we need to collect a lot of metadata and the raw data. And the other thing is the protocols. So going from the data from the experiment to the final results, everyone follows a different protocol. So collecting the protocol is, we make sure that the data can be reproduced. The R's not only stands for reusable, the data should also be reproducible. If somebody wants to check whether they did right or wrong, they should be able to reproduce the data using the protocols. So are there any plans to collect the protocols so that they can reuse the data?
Yeah, so I understand your pain. So yes, we also get the pushback on the fact that we're asking too much. And that's another driving reason why we have these kind of workshops, because it goes both ways. We want to convince you that it is useful for other people, but we also need to understand what you have identified as a bit that's hard for you. And we can then try and make it easier for you. But we don't want to back down on what we think is essential, such as the protocols, because it really is, without that information, that detailed information, nobody can reproduce your data. And you can think that that's not important, but it is not only within your lab, you want to, if you ever want to redo your experiment, but it is increasingly important for metabolomics moving into epidemiology and longitudinal studies. You need to have that information yourself, whether you think you do or not at the moment, or you think that you will remember, you won't. Okay? We know, because we see the people coming to us. So it's key in lots of different ways. Right now, we ask people to submit the protocols. We preferably ask them to upload their methods from their paper already, even if it hasn't been published, in full confidence so that we can read it. But we're also very lucky that we have 3 curators with extensive experience who have done these experiments, not all of them, but they have done experiments in the lab. So they can second guess the questions of what someone may have left out, not deliberately, but they just didn't think to, what is that? But we do know that that's part of the training use cases, ideally individual, but we do have courses that I'm sure all 3 institutes run to train new researchers and to give guidance. But all of that comes back. We need to know what the experimental community is struggling with in order to provide that training and to put as much of it into the database as validated as possible. So there are efforts in Europe with regards to the protocols. So we've built an internal ontology that if you use this machine, these are the available, and that's a lot of trawling through vendor materials, these are the available columns. So if you put in something else, you will get a message to say, and that column does not exist for this machine. What are you doing? In a polite way. Okay, but that's how we phrase it internally. So all of that needs to think, but the key thing is the database is working together. So I do know of your database existence, have looked at your spectral stuff, cool. And it would be great at some point to work together. And this is a key thing, the 3 databases represented here, we're not the only ones. As we mentioned, our Korean colleagues are here, they're setting up something similar, but also the related databases. That was what the first few slides are. There are databases all related to each other, and the better that we work together, the best. Okay, any other questions? No? Well, thank you very much for participating. There will hopefully be a white paper out of this, and definitely a workshop report. So thank you very much for participating. But please, if you were too shy to talk to us here, we are here for the rest of the week. Find us, send us a message in that cool app thing, and we will answer you. Okay, thank you. And thanks to the panel.